Hi guys, I'm back. So we have talked about DNA, replicating DNA inside the nucleus. Then we talked about how to make a protein, which is called protein synthesis. And you have transcription in the nucleus that makes mRNA, and then you have translation, which goes from mRNA to proteins. Now, what we're going to talk about today is gene regulation and mutation. Mutation is a change in the DNA. We have already done this. We probably did it when you were practicing transcription and translation. Instead of an A, you might have put a C. So that would give you a different protein, and we'll go into detail about that. Gene regulation goes back to that whole central dogma thing. It's kind of like a gene is a part of your DNA that determines your trait. So how do we know which genes are turned on or which genes are turned off? So here we go. Go back to this. Egg, sperm, fertilizes an egg, make a little baby. As that one cell continues to divide to making a large organism or to make the baby, each of those cells become differentiated. This is a review for you. Differentiated means that that cell is going to start to perform a specific job. And when it performs a specific job, it's going to have to produce different proteins. For example, muscle cells are going to produce different proteins than like your skin cells. Your skin cells can produce sweat, so they're going to have to do transcription and translation to help it produce sweat. Muscle cells don't produce sweat. However, it still has the gene in its nucleus that would allow it to produce sweat. However, it is turned off in the muscle cell, but turned on in the skin cell. So every cell in your body contains the entire DNA of your organisms, all 46 chromosomes. Then it goes down to this question. Well, then how does a blood cell look and act different from a neuron or a skin cell? If they all have the same chemical makeup, how are they doing different things? How are they performing different jobs? And that goes into gene regulation. The regulation of genes, duh. But let's look at what this means. Cells can regulate the traits they express, meaning they can turn on or off the parts of the DNA they need. So if they need to transcribe and translate a specific section of their DNA that helps it produce sweat, they would turn that on, meaning they would go ahead and transcribe it and translate that gene. If they don't need it, then that gene will be turned off. They still have the DNA in their nucleus, but they will not transcribe and translate that gene. You can use different apparatuses to help you do this in your cells. The one I want you to know is called a protein inhibitor. A protein inhibitor blocks transcription from occurring. So if you have a protein inhibitor and it's bonded to that DNA, you are not going to transcribe and translate that gene. If the inhibitor is off, which is usually like a chemical, then you can transcribe and translate that gene. There's other different mechanisms that you can use for that. However, I just want you to know that one way that we turn off a gene is known as a protein inhibitor. So this is a very sophisticated picture of what's happening. So here's transcription, here's DNA, here's mRNA. If we're making a protein, it would go DNA, mRNA, all the way to protein over here in translation. Well, if we are using a protein inhibitor, we would actually block translation from occurring. So we would actually use another type of DNA that your cell is made to bond to the mRNA so that the mRNA does not go through the second process of translation. This right here, the oligonucleotide, is a type of protein inhibitor. It will bond to the mRNA, thus not allowing it to translate into a protein. So that kind of gives you the overall idea of what happens. So this gene right here will not be expressed. This gene will not be turned on. You will not get this trait. These are fusion inhibitors, like a virus. This is something that goes on the outside of the nucleus. This is the nuclear pore down here, this little yellow thing, and it literally inhibits the mRNA from coming out. Just to give you a real life example. Okay, so that was gene regulation. Now we're going to go into another one called genetic mutations, and this is important because you'll have it on your midterm plus your EOC. This is a mutation in the gene. So let's see how this works. There's really two types of genetic mutations. One can occur at the nucleotide base level. Like I said, when we were practicing, you put a C instead of an A. That's going to be the nucleotide or the base mutation. The other one is at the whole chromosome level, and that we've actually already been introduced, and that goes into meiosis and mitosis when it doesn't split evenly or divides incorrectly. So let's look at these two. Some of these mutations can go unnoticed. I guarantee you have thousands of mutations in your bodies happening right now. Either your body is correcting it or it's not causing any serious problems. So let's look at gene mutations, meaning the ones at the base level, the nucleotide level. The first one we have is known as a point mutation. Pretty easy. This occurs at a single base pair. This is where you have one base that has changed. So like we did when we practiced, if I put a T instead of a C, that is a point mutation. So if I was supposed to have the codon ATC, but instead I had the codon AGC, that's going to change what happens after that. It's going to be transcribed differently and translated differently into a different protein. Here's a good example. So let's look over here. So we have the DNA CTT. If we look at the mRNA it's supposed to make, it's supposed to go C to G, T to A, T to A. GAA will then give us the amino acid glutamine. This will give us the normal hemoglobin, the protein on your red blood cell that sticks to oxygen. However, in a point mutation that's specific to this nucleotide base is this one over here. So for some reason, when we replicated DNA, or did the complementary bases, instead of having the correct CTT, the body puts CAT. So then we go to transcribe that, and we get GUA. A was supposed, the T that was supposed to go here was supposed to give you an A, but instead we have a U. So GUA then gave us a different amino acid known as valine. Just this one amino acid change is enough to cause what is called sickle cell hemoglobin, which is making an irregular shaped blood cell, therefore causing a disease called sickle cell. This is a point mutation. It happens at one base pair. Then there's also these guys known as frame shift mutations. Frame shift is when you either add an additional base or you did not put one there. So codons are always read in groups of threes. So regardless of what happens, your polymerase, which is the enzyme that reads the group of threes, is going to shift the DNA so that it has three. So for example, there's insertion. Insertion is when one base is mistakenly added. And then there's deletion. This is where one base is deleted and you have to shift to get back to one. Here's a good picture over here. Your sequence is supposed to be CTG, AGT, but for some reason we inserted a different base, so now we would read it as CTG, ATG. Now this would continue on and they'd still read it in the groups of three, or it might stop, it might have a premature stop. Deletion's the same thing. You're still going to read in groups of three, CTG, AGT. So you have two amino acids that would be made here, this one and this one. But if we deleted it, look what happens. CTG, we got rid of the A for unknown reasons, GT, and we would pick the next one if there was. So deletion and insertion frame shifts the entire codon from being read. Then we go into what is known as chromosomal mutations. So instead of happening just at that base level, like the ATCG, we are having mutations that happen at the entire chromosomal level. This can happen during replication, like we said, meiosis or mitosis, and it can totally alter the entire chromosome. Now, if it happens during meiosis, and that is the actual cell that is fertilized, then that child, every cell in that child will have a chromosomal abnormality. The one that we did when we did the chromosocs was the Down syndrome, and it's due to a chromosomal mutation. So what causes these mutations? Well, we don't really know. So there can be spontaneous mutations, like mistakes that are made when we replicate the DNA, when we're making the RNA, even putting the wrong amino acid there, which most of them are in our body are spontaneous mutations. We will not know why. That's what scientists are trying to do right now, is to figure out what's causing these mutations. And then there's induced mutations. Induced mutations are when the environmental agents change it. And we've already talked about these a little bit, like UV exposure, radioactivity, drugs. All of these can actually change the way you are transcribing and translating your proteins. That's the scary thing, especially it's important to teenagers right now. Your mind is still growing. You are still transcribing and translating a lot of your mental capabilities. And then now's the time where you want to explore and do all these things. Well, here's the problem. We get exposed to things like ecstasy, crack, extreme pot use, drinking. What that physically does, it can make your transcription and translation process mutate. So what you just learned that should be occurring in your body, if you go out and experiment with these things, this is actually stopping it from occurring correctly. So the scary thing is that you might not see the effects in the next couple years, but you will see the effects as your brain comes to close with growth, that you can actually see changes in your brain, which is pretty scary. If I was there, I'd tell you a story, but I can't. I'm running out of time. Okay. When we have the environment, like the drugs, smoking, chemical inhalants, carcinogenics, when we have the being exposed to our body, when we have the environment change our gene expression, it is called epigenomics. It's pretty scary. This is what we are studying. This is the cancer we're looking at. What is in our environment that is causing us to change our DNA transcription translation? And that's a whole thing called epigenomics. So I just want to say that's when the environment changes your gene expression. So what can all these mutations lead to? Well, any of these. So we have birth defects, sterility, cancer, changes in your protein, things that we don't even know yet. But then look what this one down here is. Ooh, variation of a species. So these mutations actually could be good. So if we actually have a mutation that is beneficial to an adaptation in an environment, we could have a new species. That could be a good thing. That's really the only good thing that we've seen thus far. All right, so we went through these notes pretty fast. Hopefully you got them all. And now I want to kind of fill you in with what's going on. This is my last recording, and I am expected to have a little baby boy on Tuesday. Hopefully we will be healthy. Everything is fine with me and the baby so far. He'll just be 6 weeks early, and then we will be in NICU, so I really appreciate any good vibes and thoughts that you have. So from here on out, for the rest of the semester, you should have Mr. Williams, who is awesome. So make sure that you not only have fun, but that you are learning biology. This is a really big test that you're going to take. After Mr. Williams, Ms. Davenport will be in, so it should be a smooth transition. I have changed your seating arrangement, and I have put you where I think you need to be. So I'm doing this because of situations that I've seen occur already. I'm doing this because I think you'll learn better in these seats, and oftentimes when it's not Miss Finnison there, we tend to want to play around and stuff. This hopefully will prevent that. The grades for third quarter are pretty much finalized. I put a lot of grades in there. You have your midterm exam. I hope you do study hard with that. And then in fourth quarter, that's all going to be the substitute, Miss Davenport and Mr. Williams. Miss Hartman, who y'all hopefully have met, is in room 201. If you have any questions, we are in constant contact until baby comes. But she has taken over my plans. I've had everything planned out for her. Any other biology teacher is also going to be a good source for you to tutor. They are aware that you will come to them. I wish you luck, guys. Have a great spring. Nice to get to know you, and I will see you next year. Bye, guys.